Welcome to the Evolution of Immunity module. In this talk, we're going to learn about the different protective defenses that prokaryotes have evolved. We will begin with learning about different characteristics of the immune system in higher organisms like humans, and then learn about the different mechanisms that prokaryotes have evolved to defend themselves. The immune system in higher organisms is highly evolved, and this is due to the fact that many different types of organisms are able to cause diseases in higher organisms. Thus, the higher organisms have to have mechanisms to provide protection against these different types of pathogens. The pathogens can be viruses, which we should note are non-living organisms, or they can be unicellular organisms like bacteria. Fungi are also known to infect higher organisms, and fungi can be unicellular or multicellular. Protists, which are eukaryotic organisms, can also infect higher organisms, and worms like helminths are also known to cause infections in higher organisms. Thus, the higher organisms have a variety of organisms that they have to defend themselves against. They are able to do so by producing many different types of antimicrobial compounds, as well as synthesizing different types of proteins that help in the recognition of pathogens. They have also evolved mechanisms to make variations of same type of proteins that enable in the recognition and elimination of different types of pathogens. The immune system of higher organisms also retains the memory of pathogens. Thus, the immune system of higher organisms is a product of millions of years of evolution. In order to understand how such a complex system evolved in higher organisms, let us first look at the different defenses that are present in simpler organisms like prokaryotes. Prokaryotes are the earliest living forms on Earth and belong to the kingdom of eubacteria and archaebacteria. Now, they do compete with other living organisms for space and nutrients, and thus they have evolved different types of mechanisms to protect themselves from other types of prokaryotes and even eukaryotes to a certain extent. The way they're able to protect themselves is by producing toxins to prevent the growth of other organisms. Now, here is an example where we have two different types of bacteria. And in this example, what we can see is bacteria A has secreted a toxic compound that has been perceived or sensed by bacteria B. Thus, in response to the production of this toxic compound by bacteria A, bacteria B now synthesizes toxic compounds that it releases in the environment that now affect bacteria A. And the result of that is that bacteria B's compound was much more toxic to bacteria A, and thus bacteria A is no longer able to survive. Now, with prokaryotes being able to compete with other types of prokaryotes, we should also note that prokaryotes can be infected by viruses as well. In this figure, we're able to see some of the viruses that are outlined in yellow that have infected one bacterium. In order to protect themselves against viruses, prokaryotes have evolved many different types of mechanisms. The first mechanism involves the prevention of the adsorption as well as entry of the virus. So when we look in this figure, a virus is normally able to infect a prokaryote because it is able to bind to certain types of proteins or receptors that are present on the surface of the prokaryote. Now, the bacteria or archaeal cells are able to prevent the adsorption of the virus by synthesizing micellar substances that are present outside the cell, and thus they're able to bind to the viruses outside the cell and prevent the absorption of the virus on the prokaryotic cell. Another way that the absorption of the virus can be prevented is by having certain post-translational modifications on that receptor that the viruses use to enter into the cell. Another way prokaryotes are able to prevent viral absorption and entry is by blocking the binding between the virus and the receptor. This can be done by synthesizing other types of molecules that surround the receptor and thus do not allow the virus to bind to the receptor. Another way that prokaryotes are able to prevent viruses from binding is by having other types of proteins or molecules bind to the receptor and thus not allow the receptor to be available for binding by the virus. Another way that prokaryotes are able to prevent absorption and entry is by mutating the receptor that the viruses use, and thus now the viruses are unable to bind to the receptor. Finally, another mechanism that can be used is by not expressing the receptor that the viruses use to enter into the cell. Thus, with no receptor present, the virus is unable to infect the prokaryotic cells. These are some examples, but there are other mechanisms to prevent absorption and entry as well. Another mechanism that prokaryotes have evolved is the production of restriction endonucleases. These are enzymes that are able to degrade foreign DNA like viral DNA. They're able to identify sequences as shown in the figure, and once the sequence is present, the restriction endonucleases go ahead and digest that DNA sequence. It should be noted that the bacteria and archaea have found ways to protect their own DNA so that their own DNA is not identified by restriction endonucleases and degraded. Another way that prokaryotes protect themselves against viruses is by disrupting the viral life cycle. Now, viruses that infect prokaryotes are also called as bacteriophages, and thus we can also use the term of a phage to describe viruses. Another example by which prokaryotes are able to defend themselves from viruses is by having a system called as the CRISPR-Cas system, which not only protects the bacteria against viruses, it also maintains memory of a virus that has previously infected the cell. The CRISPR-Cas system involves two components. CRISPR is the abbreviation for clustered, regularly interspaced short palindrome repeats, and it is a stretch of DNA that is present in the prokaryotes that have short stretches of viral or foreign DNA that was incorporated into the cell previously. So in this figure, the different regions of DNA that are present in those purple boxes are foreign or viral DNA that entered into the prokaryotic cell, and they have been put into the bacterial DNA. This CRISPR array that is formed can be transcribed into RNA, which is called as CRISPR RNA. This CRISPR RNA that is transcribed from the CRISPR array is called as the pre-CRISPR RNA, and it is cleaved by the Cas protein, which is an endonuclease, and this cleavage results in the formation of the CRISPR RNA. This mature CRISPR RNA is now able to associate with the Cas endonuclease, and it forms a complex called as the effector complex. If that prokaryotic cell is infected by a virus that infected it before, then we will have DNA that is complementary to that CRISPR RNA. When we have DNA that is complementary to the CRISPR RNA, then that CRISPR-Cas complex binds to that DNA that has entered the cell. And when that happens, it is able to cleave that viral DNA or foreign DNA. And thus, once the DNA gets cleaved, that DNA is, especially if it is from a virus, it is unable to infect the cell. This way, the CRISPR-Cas system is a defense mechanism against viruses. The CRISPR-Cas system has also been shown to degrade RNAs in some prokaryotes. So when we look at the figure, the CRISPR RNA that is formed can associate with a complex called as the CMR complex, and it undergoes further modifications to form a mature CRISPR RNA, which can also be called as a PSI RNA. This complex can then interact with any type of target viral RNA or foreign RNA and form complementary base pairs with that RNA, which then leads to the degradation of that target RNA. This way, the CRISPR-Cas system can not only protect the cell from DNA, but can also protect the prokaryotes from RNA viruses. With this, we come to the end of our talk, where we learned about the characteristics of the immune system in higher organisms like humans, as well as different mechanisms that prokaryotes use to defend themselves. Do remember, prokaryotes are unicellular organisms that evolved first on Earth. They're able to protect themselves by producing toxic substances and are able to combat viruses by preventing viral adsorption and entry, producing restriction endonucleases, as well as having a CRISPR-Cas system.